one of the most important lessons for a person coming from a tech background and pure software and engineering, which is biology is hard. And you don't really get to appreciate it until you hit walls. People don't know how things work for many things. And so when it comes to develop software, at least back then, for biology, usually you come and ask for specification. How should the software operate in different conditions? And the answer is, we don't know. Sometimes it should work like that, sometimes like that, because who knows what impacts a specific enzyme or protein or the RNA to be expressed. And we were like, how can you develop software like that? And that was a long journey. Together with Twist, it was a decade actually of journey of learning to be humble. And as software developers or engineers, you are used to things to move fast. You're used to get answers on the spot, and used for feedback loop to be really, really fast and iterative, because that's the way the world progresses. You have to have fast feedback loop in order to learn fast, and better to be automated, better to be using a computer, and better to be deterministic. Well, none of this is biology.
Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guests today are Yogev, Debbi, and Professor Avi Schroeder from the Technion Israel Institute of Technology, who co-founded Mana.bio along with Dr. Kira Radinsky and Roy Nevo. They're leveraging data, machine learning, and high-throughput screening to design novel lipid nanoparticles for targeted extrahepatic delivery of nucleic acid, therapeutics, and vaccines. Lipid nanoparticles are basically microscopic fatty bubbles that deliver genetic medicines into your cells. Think of them as FedEx trucks of molecular biology. Remember the mRNA vaccines that saved millions of people during COVID? They actually use lipid nanoparticles as the delivery vehicle. The little secret behind those vaccines is that we got very lucky. Those lipid nanoparticles work beautifully for the liver and immune system, but if you try to deliver RNA to your brain, your lungs, or a tumor somewhere in your body, those would be a massive failure. And scientists are still running thousands of experiments, hoping to stumble onto the right formulation. This actually was my postdoc work as well, which makes this episode very special. I always wished there was a better way to design these delivery vehicles, and that's exactly what today's guests have built with AI. In this episode of Lab to Startup, we talk about the ideation of Mana.bio, and a big part of this conversation is going to be around how biologists and chemists can interact with technologists, especially the data and AI geeks, to build something very important. This is one of my favorite conversations with guests using AI, and it also has been absolutely fun to talk to them over a period of time. And I hope you will find this enjoyable and educational as well. Yogev and Avi, I've been looking forward to this conversation since we last spoke a couple of months ago. And I think this is the first time I'm having founders from Israel on my show. Super exciting. You folks are doing some phenomenal work in the drug delivery space by combining the power of AI to design better delivery agents. Avi, maybe you can start first. What got you interested in this space?
I think lipid nanoparticles and liposomes are actually amazing. Maybe some of the most amazing particles that are out there. The reason is that they can actually travel inside the body, carry a payload, which would be a therapeutic medicine, to specific regions inside the body, and then deliver that medicine where it's needed, perform a medical task, if that's encoding for a gene or if it's delivering a small molecule medicine, which can reduce side effects and also improve the therapeutic efficacy. So in my mind, when I came across these small 100 nanometer particles the first time, I was totally blown away. And since then, I've been in love with these systems.
That's exciting. Speaking of your first time encountering these lipid nanoparticles, you publish over 50 papers and hold like 19 or 20 patents on this technology. Take us back to the moment when you realized lipid nanoparticle development was fundamentally broken as a discovery process. What was that aha moment that made you think there has to be a better way than trial and error? Talk about the state of the lipid nanoparticles then and where you are right now.
For me, the aha moment was actually during COVID time, was talking to my neighbors. Both of them were working in a tech company. They knew I work in the university, and we were talking about problems and how you solve them. They were discussing that they use data to solve problems and they use artificial intelligence. They were solving really sophisticated problems. And then they came and they asked me, Avi, how do you solve problems in the lab? And I was thinking to myself, we do a lot of trial and error, and that is not the most efficient way to go about it. And for me, that was the first aha moment, which I said, we need to change the way we develop things in the lab, the way we develop nanoparticles, the way we develop medicines. And then I needed to try and figure out how we can actually change from the ordinary way that we used to do with trial and error to a more sophisticated way using robotics and artificial intelligence. And that wasn't an easy path, but luckily I had great partners for that path.
That process of drug discovery is majority still being trial and error. We were talking before the conversation here that I was engaged in designing lipid nanoparticles, and I had a rationale of designing these things, of why a particular molecule, but then a lot of the times you still have to go through that trial and error process. If you have to describe the lipid nanoparticle formulation as historically being a black box, can you paint a picture for us when you're designing a lipid nanoparticle to deliver RNA, to say, the liver versus the brain? What are the hundreds of variables you're trying to optimize simultaneously there?
Because we're talking in nanotechnology, the first is size. The size here in nanotechnology really matters because having particles around 100 nanometers in diameter, we find enables them to actually penetrate different tissues inside the body. So you're working in really small dimensions, and into those small dimensions, you need to pack all that technology, which is the delivery technology, the targeting technology, and of course, the medicine. Those different components actually need to play different roles. For example, when you inject the particles, or even before, they need to be, when they're still in the vial, they need to be extremely stable. They need to traffic in the blood, reach that specific tissue inside the body, enter that tissue, and then from becoming stable, they need to destabilize, release their payload, and actually carry out a therapeutic task, which can be, for example, encoding for a gene. You have many different characteristics, and some of them actually contradict the others. Stability versus instability once you reach the target site. They need to be safe. All these components, those are the ones you need to actually balance when you come to design a new particle. There's so many components there that looking at it a trial and error process, you may be lucky, but there are more chances that you work that way, you'll be less lucky. You'll have an okay particle, but not an amazing one.
That aspect of serendipity in drug discovery, which people or companies don't really much talk about. We absolutely needed that, more rational way of designing drugs. Your postdoc was at MIT with Bob Langer and Dan Anderson. I mean, these are two of the legends in the field. It clearly influenced your thinking. What specific insight from that experience became foundational to Mana's approach?
Both Bob Langer and Dan Anderson are amazing people. I owe them so much. They helped shape, together with previous mentors I had, Professor Chezi Bernholtz and Joseph Kost, they really helped shape the way I look at science. What was so special about MIT is that you really believe there every day that the sky is not the limit and that through technology, you can change the world. So I think that was the first most important belief for me. Scientists can work together in order to achieve noble goals. Really, it's practiced there every day. The second thing is MIT, with all its glory, stays very humble when it comes to technology. You want to reach the highest goals, but you understand that at the end of the day, there's a patient that is really waiting for our technology to come and to save their lives, improve their lives, treat a condition. We kept that in mind every day. It was that blend of being very scientific, but also being practical and trying to save the world. I would say that really is one of the things that drove us.
So this is so lovely to hear. This intentionality you're talking about. It's just not research for the sake of a paper or a patent, but there's this end goal and the aspect of working together, which a lot of times I think is missing in academia. Most of the people are so siloed in their own labs. I see it with grad students and postdocs. I keep telling them, you need to work with other people. But Yogev, you had a fascinating path from being a chief operating officer at Genome Compiler, which was acquired by Twist Biosciences, to leading the Twist Israel R&D through the IPO, and then a stint at Mana.com during the IPO. This is a fascinating ride. What was that thread that pulled you from software into the life sciences?
Actually, if we go back to the history a little bit, after graduating from computer science, I actually worked at Intel Corporation for many years. So the software programmer, team manager, program manager in the software group. So nothing to do with bio indeed. That was a coincidence when back then, together with my business partner and longtime friend, Roy Nevo, we worked together at Intel and we met Omri Drori, which was back then postdoc at Stanford. We're talking 2010. He came with an idea that we have to build a software for gene editing, or actually DNA editing, which was just started, the whole DNA synthesis field. We were enthusiastic and we said, sure, sounds like a great idea for a startup. So we did the Genome Compiler together. And then I learned, I think one of the most important lessons for a person coming from a tech background and pure software and engineering, which is biology is hard. And you don't really get to appreciate it until you hit walls. People don't know how things work for many things. And so when it comes to develop software, at least back then, for biology, usually you come and ask for specification. How should the software operate in different conditions? And the answer is, we don't know. Sometimes it should work like that, sometimes like that, because who knows what impacts a specific enzyme or protein or RNA to be expressed. And we were like, how can you develop software like that? And that was a long journey. Together with Twist, it was a decade, actually, of journey of learning to be humble. And as software developers or engineers, you are used to things to move fast. You're used to get answers on the spot. And used for feedback loop to be really, really fast and iterative, because that's the way the world progresses. You have to have fast feedback loop in order to learn fast, and better to be automated, better to be using a computer, and better to be deterministic. Well, none of this is biology. It was hard, it was rewarding, it was fulfilling, like amazing journey. During COVID, both Roy and I felt that we had done like great. We worked at Intel, our first company got acquired. We participated in 2 IPOs, like you mentioned, Twist and Monday. The companies went great, like Twist in the IPO were $2 billion, Monday at the IPO, $10 billion. And we felt, it's time to have a new company, which might be bigger. One thing that guided us, except of it has to do something with engineering, with software and AI, because that's our background, we want to do something impactful in the life science space. We got hooked on that matter. So we got hooked. We want to do something which will change lives, will contribute to better human health and better quality of life on Earth. So then during COVID, we went for ideation phase. The two of us with our background, we knew what we're aiming. We didn't know what field it's going to be in. And so we have talked with many different super smart people, from scientists, professors, entrepreneurs, investors, business people, doctors in different fields. My wife is a dentist, so we interviewed her. I have an uncle, which is an orthopedian, so we talked with him, and we just looked for the best idea. And through this network, through the network that we have built, we got to Avi here. He is an amazing individual. You just got it. And we immediately fell in love, both with the idea and with the person.
This is great background. Thank you for setting that up. Maybe you can talk about when you and Avi first started discussing ideas. What was that specific problem statement you wrote on that whiteboard? Was it we need better lipid nanoparticles, or we need to turn drug delivery into a data science problem? Walk us through that discussion happening between you two.
Roy and I, we went through this ideation phase. Someone within our network connected us to Avi, and then we came to Avi. We told him this story. We said, this is our background. What we want to do is something which will leverage our capabilities and experience. Software, AI, building companies, operators, entrepreneurial, and we want to do something impactful. We want to do something in the life science space. So we heard you have many great ideas and multiple companies out of your lab. So what do you think? Do you have any idea? So that was the beginning.
This meeting was end of the day, and it's one of those meetings I actually hope. So I didn't know Yogev and Roy. I was connected through Omri, who's an amazing guy, really an amazing entrepreneur. It was like around 5 or 6 p.m. And I remember calling them, hoping maybe that the meeting would not take place and I could go home a bit earlier. So it was one of those long days. And I call and I hear the energies in the car. Yeah, we're on our way to Haifa, to the Technion. I say, okay, let's go down to the lab together. And instead of sitting in the office, we went down to the lab and I told them about this project that we just began. And it was actually Professor Kira Radinsky, an expert in computer science, one of Israel's really leading scientists in that field, who also understands artificial intelligence really deeply, started working together with our group where we were trying to combine AI in order to better design lipid nanoparticles. This all started from that discussion I had with my neighbors because I could not do it myself. And Naresh, it goes back exactly to the point you said. I'm a chemical engineer. I did not know how to do robotics and I did not know how to do artificial intelligence. The robotics we were able to learn, but the AI we just couldn't do ourselves. A very talented student from my lab worked with a very talented student from Kira's lab. Within a very short period of time, we started seeing amazing data, things we've never seen before. Because instead of being limited to the materials, the chemicals that we usually use in our lab, they were bringing in data and actually suggesting totally new molecules in order to design lipid nanoparticles. We started developing new lipid nanoparticles and we saw, wow, this is working better than we expected. And that's exactly the point where Yogev and Roy came to visit our lab. I walk around, I show them the robot, and their eyes light up. They say, this is exactly the problem we were looking for. It includes biology, engineering, but also sophisticated computation, and that's really their strength. For me, I will say it goes back to that point that you mentioned, working together. It's Kira Radinsky, and also management skills together with the knowledge which Yogev actually brings and Roy brings. So building a full team together with our labs and now the company, of course, Mana, it all works together. And it's people from different disciplines. So for me, every day is learning.
It would have happened if Yogev didn't walk into your lab and you started talking about what was happening. This is the core of what I really, really want to be doing with the universities. I think more of such interactions need to happen. Thank you for sharing that story of yours.
On our side, when we came to Avi's lab and he showed us around the lab and the science and the story, one of the major things that attracted us to this journey, and I think this is a good takeaway for all the scientists that are listening, is that Avi not only understood the science to the atom, to the bit, and was able to explain to us in simple words for us, but also he was able to make the compelling business case. So he understood the market. Not only understood the science, he also understood the market and the gap and the demand in terms that not only convinced me, but I was able to understand, okay, this is, and we were looking for scientific co-founders, Roy and I, because we're not coming from the science field. And I said, okay, this is the person that I can take to business meetings, to investors meetings, to build the investment pitch with, because he really understands the market. And from there, of course, we need to learn much more, but this was a really strong kickstart.
MIT, when you enter it, it's about not only developing science, it's about implementing it and making an impact. And that cannot happen only through research papers. It happens through the translation, taking these discoveries, making something out of them. That resonates with many of the people in MIT. And you start speaking that language, because we speak one language in academia and people in industry speak a different language. And sometimes that disconnect in language can actually be harmful to great technologies that could help many, many people. I think we need education from both sides. We, the scientists, need to be educated how to speak with people who are translational experts. They're experts in their field, and the other way around too.
Absolutely. I mean, speaking of language, maybe Yogev can fill in some more story. You talk to so many other professors and scientists and top people in their space. What was the language there? What is different in those conversations compared to what you had with Avi?
So I think as a scientist, when you work on such difficult problems, you fall in love with the science. And one thing that people sometimes miss is the business case. So that was a really enlightening moment that we came to speak with yet another quote unquote crazy professor, someone with lots of ideas, with deep understanding. We weren't expecting getting reasoning for the business case. So we got that. So that's A. B is ability to explain the science using simple high-level language without the urge to go deep into the science and just dig into all the details in order to impress someone. For me, I'm not impressed with more data, more details, more validation. I'm impressed with simplicity, explainability, and coherent story about the business case. Then when we ask questions, we got answers that made us believe, okay, there is like the depth that is needed here. This is really validated, backed by papers, etc.
Simplicity and explainability. Those are great takeaways from this little conversation. There are a lot of things happening in academia where Nobel Prize winners are starting companies. Jennifer Doudna, Caroline Bertozzi at Stanford, and so on. People are realizing that. I hope more of them pick up on that.
Avi is a chemical engineer by training, and is one of the best. But another advantage as a scientist that you can have is ability to understand the other fields and the way they can integrate with you. So the interdisciplinarity of your environment is crucial, especially today when you can actually combine different fields. Listening to Avi's perspective, he got this idea from totally different area, totally different field. He immediately understood how he can apply it to his field, the problems that he sees with the science that he is the owner. And so I think this is super admirable. And that's what actually led to Mana, which is a real integration between the technology, AI, software, robotics, into chemical engineering and to this specific scientific problem.
Absolutely. You're in the software AI space, and you call your platform Mina, which has been called the ChatGPT for lipid nanoparticles. What is really happening under the hood? Are you training foundation models on chemical structures? Or is this more about multi-model learning for literature, experimental data, and molecular properties? What is happening under the hood there?
Mina, the ChatGPT for LNP, is a different product. It's just another tool that sits on top of all the data that we have built. The platform itself is built upon two main data sources. In order for AI to work, you need data. And the two main data sources that we leverage, one is from the literature and the other one is from our own lab. On the literature, we have actually leveraged the 20 plus years of research. So we developed automation tools that go and scrape the entire literature, papers, patents, publications, everything is formatted, curated, normalized, and uploaded into our trainings. We developed software tools that automatically collect everything from Google Scholar and other databases and scrape the actual PDF. We did it way before ChatGPT. So today you can compare what we do with ChatGPT. Ours is much more fine-tuned into this scientific space and specifically for lipid nanoparticles. We harnessed everything from molecule structures, charts, images, text, everything we normalized into our data. So that's on the publication side. It's not enough. Why? Because if you think about it, albeit 20 years of research, A, there is limited success, and B, people do not publish everything. You do not publish toxicity data.
You basically publish the best result, and that's what you do.
Only the best results which are not always repeatable, but that's another point. But we need negative data for AI. In order for the AI to really need negative data. And not everything is published. This is why we also set up our own lab. So we invested in equipment, in materials, and of course, a full lab team that is responsible for generating our own proprietary data in our wet lab. And by that, we close the loop. So we really close the loop between AI and lab in a way that the whole goal for our lab is to generate data. For us, it doesn't really matter if an experiment by scientists works or doesn't work. On the contrary, it's good if it doesn't work, because then we generate negative data, which we lack from the literature. For us, what's important in the lab in order to generate data, we need to make sure that all the protocols are being followed as they are. We need to make sure you use the same materials, and we need to make sure that all the data eventually is actually uploaded to the database after doing some QC, quality control, etc. So in order to make sure that all these steps are accurate, we build software tools that track the data flow and making sure that all the steps are taken, and eventually the data is uploaded to the training set. And in fact, our scientists are measured by the amount and quality of data points that are generated in the lab. We don't care about the end result. All we care is whether the experiment was done properly, as expected, and then we know that we can trust the result.
A big part of it looks like cleaning up a lot of data that you find, which is very important.
In the literature, you need to do a lot of cleanups. Not only cleanups, by the way, we also invested a lot in, for example, normalization of the data, because on published data, people use different controls and different protocols and different assays. So you need to know how to really normalize the data properly and how to clean up outliers. When you look at the big picture, sometimes there are points that doesn't make sense, too good to be true. We take these data points out. So we have a team that uses our own automation tools, but also kind of review and curate the data. In our wet lab, the challenge is really to streamline the process, meaning develop our own software tools, as I mentioned, but also making the people working together and understand each other. The example that I can use is exactly the experience that we had in our previous company, which is software engineers, computer scientists, you use for things to happen in a certain way. And when they don't, you get mad. What do you mean it doesn't work? It should work. But biology is not engineering, so it doesn't always work, and it's not deterministic, and we don't know how to explain it. And one of the things that I keep telling software engineers and people from tech background is, while it's software, it's human-made, and we know the underlying logic and language, and we have documentation and everything, there is always an expert which can explain to us any library or whatever we use. In biology, we don't know how it works. God did not give us documentation for the code, for the DNA. So we don't know. So it's an iterative process, and people need to learn how to work together. The best people in the company, and the best people that are, I think, the best candidates for such a company, are people that have deep expertise in their field, but they can understand, appreciate, and work with people from other fields. That's where the magic happens. When people from different fields really working together.
With an open mind and be curious. Avi, maybe you can step in here. How big is this team? What is happening in terms of just reproducing data, and what kind of automation went into that? If you can set the stage for that.
I'll actually start from the wet lab experience. I've been working in a wet lab for so many years, and we've been even trained to think that it's an art, and you start becoming an artist where you can design your particle, and you're probably the best in designing that particle. I was even a bit skeptic. Could this computer really outperform us as humans? Not only myself, many of the experts in the field, Yogev and I, when we started Mana, unfortunately, we didn't record these discussions, but with very prominent scientists, they were tolerant of us and respectful in front of us, but we knew that they were also very skeptic, and they told that to our face. And Yogev and I, we decided we're not going to give up on this dream. Slowly, both in the lab, we started generating this new data. There's no failed data. It's that data that is not out there. Together with the data that's out there, we started seeing amazing results, targeting of the particles to specific regions inside the body. I think that a lot of the data is reproducible that's out there, because I know my colleagues I trust them. And it's true, a paper may come out where things may not be exactly reproducible, but to say that the majority of data is not reproducible, I don't want to think that on scientists. I believe scientists, I believe in science, and I think as such, I also believe that the research body that's out there is true. The example I give myself also is that when I use ways to drive from Tel Aviv to Haifa or to drive to work in the morning, I know that there is a car that it has Waze on or Google Maps and may stop on the side or may take a different turn. But the general population, the data is being built and you get a pretty good picture. And I think the noise does not affect the major results we have out there. One of the major things I think is the question is where there's no data. How do we generate data in areas where there's no data? It's total new innovative spaces such as targeting and new biologies that are being discovered. There, generating data is a main challenge. And inside the lab, it's a crosstalk between the data scientist and also the web biologist and chemist that are there. It took a couple months until the people really understood. And it was nice to see how over the time, people that come in now, they start speaking a joint language. It's a new language. It's a mix of the language of computer science and robotics together with the language of chemistry and biology.
You had a lot of data that was published. If you think about the data modes, you seem to claim you have the world's largest curated lipid nanoparticle data set. In a field where most research is published, what makes your data uniquely valuable? What is proprietary versus that in the public domain here so far?
When we develop those bots that read the published papers and actually bring in some of that successful data, they were looking, for example, biodistribution information. You have many times an image of tissue where the particles actually accumulated, for example, in the heart or the lungs or the kidneys. But that data is not actually data that you can come and pass it on and run computation on. So our bots actually will read that image, change it into a data file, and that data file now can actually be analyzed. And the same is also for graphs that are published in many of the papers. The unique side of the data we have is looking at data that people did not publish. For example, during the development of a particle, there are many, many steps that people do not mention. They'll just write it in a sentence, but in fact, it's probably 1 or 2 years of development to reach that particle that is stable. And that's what Mana really has inside. It's all those steps exist inside our database because we measure almost every aspect of a nanoparticle that we develop. We'll measure its size, of course, and its zeta potential, but many, many other characteristics of each one of these nanoparticles in order to make sure that it performs exactly the way we designed it to perform and based on the target it has inside the body.
That's amazing. Maybe you can walk us through a typical week in your lab. How does AI suggest a few formulations? And how quickly can you synthesize and test them? And how does the data feedback go into the model?
The amazing thing about AI, and I'm quoting here Radinsky, is that the actual number of data points you need to have has not been figured out theoretically yet. Meaning that if you have great data, you may not have to have hundreds of thousands of data points in order to build a very strong model. When I look at AI, it's not about high throughput anymore. It's about running smart experiments where you get the right data points. So in Mana, we were able, through very few animal trials, reducing the amount of animals we run in the trials, which I think has ethical value. We were able to reach the same goals that our competitors may have run through tens and tens of preclinical animal trials. Through the design of AI, you're able to run more sophisticated studies by asking the questions in a way that you can actually reach your point with less experiments. And I think that's the great part of it. So you're almost less wasteful. The computer is actually doing that extra work instead of lab work.
3 points. About the data mode, I'd like to emphasize maybe at this point the Mina that you mentioned earlier. So one of the things that we have done after we got all the public data out there digested into our training set, we have decided to add on top of it an LLM-based AI agent layer to make this data accessible through simple queries, really, like a chatbot. When we did it, and we also added our own proprietary data, suddenly it had amazing impact. Because suddenly you can simply talk with all our data that was established throughout the years, and being able to analyze the data, get insights on past experiments that you already forgotten, troubleshoot, and even ask the AI agent, what should I work on next for the next week? That had amazing impact on the team. And then we said, why don't we publish it to the community to accelerate science all around us? The proprietary data we left only for the internal version. And then the Mina, the ChatGPT for NLP that you see now on the website, is only trained on the public data. So now what you can do is to ask the public data, and the answers are real. They are based on all the 20 plus years of research that is out there in the literature. And you get real DOI, real reference to real things. So there is no hallucination here. And this is really the intention, is to accelerate science all around us and to show also the power of Mana's AI team. And I just want to shout out the Mina team within Mana, which done an amazing job, and everyone is welcome to use it. It's free on our website. The other point regarding data mode is the way you represent data. Avi touched upon it a bit. In order to use AI for scientific data, you need to have some way to digitalize it, to represent it in some digital representation, mostly text. There is a way to represent molecules. It's called SMILES. People use it. We didn't invent it, but there is no representation for a full LNP, full lipid nanoparticle. So that's one thing, for example, that we need to invent and then use it, in addition to all those automation scraping tools that Avi mentioned, digitalize the charts and the images or the accumulation of the LNPs. So this is another point within our data that is part of the data mode. When we started the company, it was clear to us, Roy and I, when we did this ideation, we knew that we need the deep scientific co-founder. Lucky us, we got both Avi and Kira. And the second part was, we knew what culture we want to have in the company. We actually drafted the culture way before we knew what the company is going to be about, because Roy and I are working for the past 20 years together, so we know how we want things to operate. But the next thing, we knew it's going to be some sort of a tech company, in a sense of the way that tech company works. With all the ceremonies, with daily stand-up and sprints and retro and demo, and those of you in the tech industry know what I'm talking about, the agile and all of. And so when you come to Mana, you see on one side an open space with like screens and dashboards and typical high-tech startup scene. But then you suddenly see a lab, which is top of the line lab with all the high-end equipment and everything. So this fusion is the magic. And the weekly life at Mana start with weekly kickoff ceremony where we go project by project and each one kind of explains what it's going to do. We have daily meetings, and we do the daily meetings next to the dashboard, and we talk about the metrics of the last day or last week that needed to touch upon. These dashboards show and elevate everything that is going at Mana, from what experiments are being run, what data points are being generated. In fact, in the lab, they have their own dashboard that shows expected data points versus actual data points that were generated during this week. So every week we generate hundreds of data points. We have metrics that shows even budget used, so people are aware of the budget and how much it costs to run experiments. And we do it not in a notion just to save money. We do it in a notion of maximize the capital efficiency that we work upon. Avi touched upon the fact that we save lots of animal lives, and it's good ethically, but it's also good financially. It's also good economically. Because once you can use these technologies, AI and data science and machine learning models, and the fact that you have this hybrid in the team, you can actually get to results faster. So you use much less animals, much less money, and most importantly, much faster and much less time. And this is the point which I'm proud of the most after the team.
These are two different worlds of startups coming and working together. Maybe you can talk about the cross-functional team dynamics. Here you have on the AI side, speed is so important. Things have to move fast. And then we have our limitations of the biochemistry side. Maybe you can share some stories of clashes between these two teams, the expectations were not set right, and how you resolve those things.
You're correct, Naresh. This is like running two startups within one startup, which makes it fun and challenging at times. And exactly this is one of the realization that one can understand working with these two group of people. Different cultures, different language, and different backgrounds. Now, if you think about it, when you start a company, you get experienced people. In the tech world, you're exactly right. People are optimizing for time. You want to get things fast. The competition is fierce. You have access to capital. We see what's going on recently with all the AI hype or whatever. You get lots of funding. You move fast, break things. This is a saying. However, on the science side, on the biology, chemistry, people are tuned to optimize for money, not for time. Why? Because things take a lot of time anyways. To get drug to the market, it takes 10, 15 years and billions of dollars. As an individual, if you're about to save a week or 2 weeks or even a month of work, it's meaningless almost. So this is not super important. But on the other hand, money is really short. And there is an interesting kind of observation. The best people in the pharma industry, they grew up in many years in the academia, PhDs and postdocs. So you spend more than a decade in the academia, which is, if you think about it, it's like the greenhouse for this culture, because in academia also, you don't have a real sense of urgency, which is, by the way, intentionally, it's good, because then you have the labor to do creative science, and we need it. So I'm not criticizing it. It just this is the way it is. And then money, you're short of money. So you learn how to save money. So when we started the company, we were like, okay, we need to move fast. We were lucky to raise when we started, because it's not our first rodeo. So our investors from Genome Compiler backed us immediately, NFX and Lambert. And one of the things that I realized coming to the lab in the first weeks was that one of the scientists, which is super talented, he was working on optimizing some process that would result in saving a lot of money, but it was only him working on creating new assays, and instead of working on creating new assays, he just worked on some side project to try to save us some money. That was not significant. It was money, but not significant. I remember coming to him and learning about what he does, and explained to me and said, oh, this is not what you're trying to do, but do you really need it? Because we do have the money to spend. It's budgeted. So we know it's okay to spend it. And I need your brain to work on these things, which will help us move faster. And it was hard in the beginning to get it, but eventually got it. That was an enlightening moment. And I think the team quickly realized this fusion that needs to be happening. Another example is on the other side, when one of the software engineers get into the lab. Initially, we needed to push the software engineers to get into the lab. It's not natural for them. He went into the lab and he sees the chemist. And then the chemist comes out and he starts to type things on a spreadsheet. He was making a really mundane work of typing different molecules on the spreadsheet. And the software engineer is like, oh, what are you doing? And then explain. And he says, are you going to do it manual? Like, imagine typing similar words on a spreadsheet, word by word, and doing it 1,000 times. And he says, yeah, what else can I do? We need this list. Yogev asked for this list. Avi is waiting. The software engineer is like, well, I can automate it. It's a simple script for me. So he did this script, and in half an hour of work, he saved like 2 weeks of work for the chemists. I have these examples where you see this is where magic happens, when the two worlds really combine and working together.
These are all real problems. I was laughing earlier because when you said the scientist was trying to save a bit of money, that is how we are trained in the lab. Every bit counts, and changing that culture. How do you measure time impact versus money impact? That is something that people without money might not have. I want to dig a little bit different. Maybe Avi, say when your postdoc says like, hey, their model is overfitting on the lipid class. And Roy's engineering team says we need more training data with better labels. How do you bridge that gap? At what point did you realize that, hey, we need some frameworks or process. What did that evolution look like? And what do you guys do? Give me 2 examples of what really helped that comes to your mind.
I remember numerous funny points that we had in the beginning of Mana. It's the two sides talking. And the people of computer science, they have a good sense of humor. I used to, many times when we call the people, I used to say, Rabotai, that's the Hebrew word for gentleman. In the tech world, they must not be using that word for some reason. And they were so astonished that they actually called one of our software tools Rabotai. So they took for one role and they brought it over. I think that what really connects us all is at the end of the day is a real passion. The people that come to Mana, also the computer scientists and also the chemists and biologists, they all have the same passion to improve our world, improve medicine through technology, because you need a lot of patience. It's much easier to work either in a tech company or in a chemical or biological company. Working in that merged region, which I think is very, very fulfilling because you learn so many new techniques and traits. Every day is new, but it also takes a lot of patience. And there are moments that it's not easy. And we've also seen throughout the years, some of the people, it's harder for them to adapt to actually this duality. And they choose to go back to one of the sides, either all chemistry, biology, or all computation. But we did try to bring people that do speak that passion, at least. There was not a lot of belief in computation, I think, when we grew up as scientists. I think AI changed that dramatically. I say doing science without artificial intelligence is like driving without a GPS. You can do it. And you may also reach your target site. It's just a lot less efficient. When you actually use Google Maps or Waze or what other tool that's out there, you're more efficient in what you do. And for me, it's today the only way of actually doing science. Computation also, or at least its implementation inside science, really made a very, very big advancement throughout the past years, which allows this dramatic change in the way we implement it. And the way I look at it is from a technical perspective, if the models, which some of them are extremely accurate, but some are less accurate, they look from the beginning to the end. AI looks from the end to the beginning. So it looks at all the aspects that bring the result, and they try to correlate between the two. It seems for super complex problems, which have multiple variables, that the artificial intelligence way of looking at these problems, at least in my experience in Mana, gives a better result than predictions that people are trying to do and still are trying through models alone. And maybe the future will also bring models into artificial intelligence and really reinforce one another. And I think that will be a total disruptive technology. And we're already starting to see the first examples of that, where models of known and well-established chemical and biological phenomena are brought into artificial intelligence in order to strengthen the models that we have there. It also saves some computation time, but it also strengthens the AI models that we have there, especially in areas where we lack data.
When your AI suggests a completely novel LNP formulation that violates conventional chemistry wisdom like you and I deal with, how do you decide whether to trust the model or the domain expertise? Have there been cases where AI was right and the chemist was wrong, or vice versa?
Totally. Our main chemist, our chief chemist in Mana is called Ego. That's his name. And I remember the two of us looking at a molecule and a formulation, because Mana does not only design, for example, the ionizable lipids that are there for compacting the nucleic acids. Mana actually looks at the entire formulation, all the lipids that construct the lipid nanoparticle. And we were looking at one another and we were saying, can't be. This molecule will never work. But I forced myself, and I told Yogev when we started the company, I said, Yogev, listen, when I start using my human intelligence and overriding the artificial intelligence, please stop me. Of course, it's natural to say, I know this molecule will not work. And Yogev was very careful about that. And I was also careful. And we tried that molecule and it worked amazingly. Sometimes the computer, because we bring in so much information into it, actually sees things that we don't see. And it's not only us. I remember us sitting, one of the first big meetings we had with a very large pharmaceutical company. We're talking chemists that have each one 40 or 50 years of experience. And they all came to sit with us. And what they did is they gave us a list of 10 molecules. And the 10 molecules, they asked us to rank them for their ability to deliver a nucleic acid. And we, of course, ran no experiments. We also would never have had time. We ran it only in our model. And I'll tell you the truth, Naresh, I was sweating and I was hardly breathing because we didn't know. We ranked them according to the model.
Yeah.
I heard the quiet also from the other side of the line. Their jaws dropped. When we put molecule after molecule, I think we missed in the middle, maybe 2 that we switched their spots, but 8 out of the 10 were point that, and they gave us hard molecules. I'm telling you, these are molecules. They'll change one small chemical group here. You hardly see the difference. When you look at the molecule, you have to look for a couple seconds to see where the difference is. And the computer saw it immediately. For me, that was the first proof that we're on to something. Today, fortunately, not only we recognize that, we see scientist after scientist, people are advocating for this use. And I think we were early on, but it seems to me that everyone is aligned now to this use of or integration of AI into our workflow.
Yogev, how about you? Did you see anything that you proposed that was wrong, and what did you learn from that? And how do you temper these teams?
So you can see it as a contest sometimes when you have a molecule that the app suggests, or the chemist, and then the AI, and let's see who is better. So the point is that the AI aim to help science. It's like driving with the GPS. We actually call it AI-assisted science. And we need to be humble. We don't expect the AI to be absolutely 100 times better, and that's it. No, it can't be, because you don't have all the data out there yet. What we do to compensate it and also to leverage what we can to generate data that will be sufficient enough for the training set, you actually build your wet lab data generation process on top of really diverse set of recommendations. So let's say the AI gives you a list. By the way, every week we do this exercise. You have hundreds of millions actually of permutations, and the AI rank according to what you wanted to do. So if you work on some physchem, so things like a size of formulation, so we have a few weeks of working on size prediction or toxicity prediction for different cytokines or specific cell targeting. We have kind of a list of recommendations by the AI with the top ones to be, okay, this is supposed to work, and the really bottom line in the don't supposed to work. So what we do, we actually take a blend. So we take a blend from the top, we take a blend from the bottom to get negative results, or to see whether the AI is correct, or to generate more data. And then we take some random from the middle, and in the random, we take whatever the chemists suggest. And that's how we balance the tension. We anyway need to generate data, so let's do it. And at times you see the AI actually outperform the chemist. Sometimes the chemists give a better kind of intuition. It's a good exercise. I think the name of the game is to generate this and being able to learn from it. So being able to train the model. We have dashboard that shows the accuracy of the models that are getting better in time. By the way, sometimes there is a bump. You see it getting more and more accurate, but then there is a bump. Why? Because suddenly you introduced a new structure, a new chemistry, which the model didn't know because it wasn't trained on. That's good. Now we have a new sort of chemistry. One important point to emphasize on that, because you mentioned what happens if there is a new structure and you don't know how to make it. So we make sure that all the structures that the AI model actually predicts are synthesizable, manufacturable. When we started, it was just really strict and limited to known parts, cheap parts, available parts, building blocks that you can make the molecule on with known chemical bonds. You cannot have the AI too creative, because then it might be too expensive or even how to make it. Now we also baked in a full synthesizability model to really take it a step forward to make sure that it is not only synthesizable, but also looking forward to make sure it will be scalable to scale up once we get to CMC.
You bring up a good point of the learning is going on all the time. Your AI is only as good as your training data. But then what also comes to my mind is biological experiments, they are messy. They have batch effects, and sometimes they just fail. Tell us about a time when bad experiments experimental data led the AI astray. How do you diagnose that? And what systems do you have in place to catch such phenomena?
At the end of the day, we always remember that we have a human. One example, which we all can relate to, is that, if you ask ChatGPT a question, you'll read it sometimes and you say, no, that's wrong. That may not be the right result. And that's because you're asking something the system does not know. And for us, when we started, it was also about robotics and generating data, and also making sure that data is curated in the right way and the tables actually pass the data into our models in the right way. And there were a lot of glitches in the beginning of Mana. It's because it's a learning of everyone in the system. It's how the results are actually transformed, what time you take the result, and teaching people two different things, especially when you record that data point, is really extremely, extremely important. How you record it and making sure that the conditions are actually exactly the same, the way you record that data point between each and every cycle, because if not, it's hard for the system to compare between one experiment and another and to actually learn from all these. And we probably had in the beginning several experiments where we learned the hard way. We said, oh, that data is shot because we took it at a different time point. We used a different measure. It was actually training our teams to think in a way of data rather than a way of results. And that took some time. Also, the robotic systems that we use today, as sophisticated and good as they are, these systems are not failure prone. Sometimes the machine may not pick up a pipette. One of the liquids that we use or the media may run out. Those things can happen. Catching that early on to make sure we have a problem here and not recording that data is extremely important. In the beginning of Mana, we actually needed to build software that overlooks our computational process in order to make sure that we don't have those errors. But we only thought to develop it, and that's part of under the hood, which you mentioned. That happened only after we understood we're having from time to time problems that we don't know, we're not aware of. We're getting suddenly a set of data that just doesn't make sense with anything else we measured. And then you go back and you check and you say, oh, we actually made a mistake in the way we ran this experiment. We didn't use the right media. We didn't use the right readout or time points, etc.
Just to add on that, a small example. One time in the lab, one experiment didn't work as expected. It wasn't only as expected by the AI, it was something that wasn't produced from previous runs. And that doesn't make sense because we have the protocol, we know what needs to be done. Repeated over and over, it was a formulation or set of formulations that needed to read the results just to make sure the QC works before the next step. And something didn't work. And you know how it is. You repeat it, you change things, you try to change materials, whatnot. Eventually, the root cause was that one of the materials actually expired. It's something that you're supposed to know when you run experiment, but it's easy to spot when you do manual work and few formulations in a run. When you do hundreds, you can't really track it manually. One immediate retro to that is that we need some sort of automation to all the inventory that we have for expiry date. So we baked it into the software, which called Robot. It's also kind of a reference to robot AI. So we actually baked into the system, we developed the automation alert system to alert whenever an inventory is going to be expired. So we know not only not to use this material, but also when to order in advance. So we never shop. And one of the things that we do, and that goes back to how we work at Mana, is that at the end of every week, the closing meeting is demo and retro. So from the tech world, like everyone brag with what he or she achieved this week. So showing whatever experiment results or new feature in the software tool. And then retro. Retro is retrospect analysis, what we did good, what we did not so good and need to be improved. And this is how we improved, continuously improvement on a weekly basis, the way we work at Mana.
You're making me want to come back into the lab. These are the things I missed going for the 96 wells. There's always going to be mistakes and just automating the process. This could have been magic if it existed when I was in the lab. We learned, it's all fun. Let's shift the gears and talk about the business model and strategy for you guys. It looks like you're launching Mina for free to the research community, but keeping some of your most advanced models proprietary. This feels like the freemium SaaS play applied to biotechnology. What is your long-term play here?
Mina is just some kind of showcase tool. You can look at Mina as giving back to the community. At the end of the day, all the data that Mina is trained on is public data that was published by you guys, by you and Avi and other great scientists. So that's how we look at Mina. That's not the business model. So the business model for Mana, I'll start with the problem statement. To make the business case, the genetic medicine today holds the largest promise ever. It's clearly the next generation of therapeutics, vaccines for oncology, immunology, rare diseases, and others. But the one main limiting factor is delivery, as we know. Every delivery solution is different. Why? Because you need it to be highly customized to the specific payload, whether it is messenger RNA or CRISPR or DNA or ASO or different editing payloads. It has to go to the specific cell type in the tissue. There might be different behavior that you would require, like circulation time, things like that. And then on top of that, obviously, it has to be highly optimized for safety, for manufacturability, for tolerability, etc. That broad set of gaps in the industry are actually opportunity for a high demand for platform for delivery. So what we have built at Mana is this platform that can actually serve all the different tasks, all the different challenges, all the different permutations that are needed. And that's the vision. The vision is to build customized solutions, delivery solutions for your need. So the business model is working together with our partners, which are experts in the biology, identify the target, the indication, and designing the actual payload, working with us to tailor the delivery, and then license it out to our partner. So at the end of the day, the business model is a classic, traditional biotech licensing model where we are responsible for the delivery molecules, we are responsible for the LNP, we own the IP, and then we license out to our partners in the traditional deal of upfront milestones and royalties.
Do you see yourself growing into a therapeutic company, like a Mana Therapeutics, one day?
Yes, definitely. And we are on the way to eventually dominate this. Once you have this powerful platform, then you can start playing with not only out-license the delivery solutions to our partners, which can then sponsor the preclinical and clinical development and take it to commercialization. You can actually get into a position that's our plan to in-license payload and sponsor the preclinical and clinical development ourselves. We're starting from a pure platform play in order to gain validation, to get to a commercial product, in order to get also education about the process, non-dilutive funding, of course, from the pharma partners. And then at the next stage of Mana, we'll be well positioned to take some of our unique delivery capabilities and actually enjoy the value of the economics of in-licensing payloads from partners and then take it ourselves to the clinic.
I don't think I ever thought much about the regulatory pathway there. How do you think about the FDA validation of AI-designed drug delivery systems? Does it exist? Do you need to prove the AI reasoning or just the safety and efficacy of the end product? How does it work?
The way I look at it is that the FDA does not really care, where the molecule came from. You can dream it or you can invent it using artificial intelligence. It does care very much that it's safe and efficacious. And this past year also, they are signaling that they are looking also at the process for the first time, I think in many, many years, pushing to more models that can actually develop drugs more efficiently. Two things are happening. One is that genetic medicines are entering, and we understand that they'll differ from one patient to another. Some will be good for large crowds, but others will have to be good for a specific small population. And for that, we need new tools and also a new way of looking at the regulatory pathway. We will not be able to run the full approval for each type of particle that we have. But if Mana, for example, develops a nanoparticle that reaches the lung, and one patient will treat lung cancer, and for another, we may treat lung fibrosis, I think that actually makes a lot of sense. And again, we'd have to look at the biology behind it, the safety behind it, but it would be a great starting point, I think, for treating patients in a more personalized manner.
I'll add that in the regulatory path, as Avi said, eventually you need to prove the data points themselves, safety, efficacy, etc. The AI could be an advantage because what they want to see eventually are new ways to save animals and time. So that's an advantage. Another point on that is that not only the FDA cares about the part that AI is behind, but also our partners. That's actually a challenge. I think it's a challenge for all the AI and bio companies that are now growing, which is the way that you got to this molecule. The data is not convincing by itself. Like even if you show beautiful data, which is best in class and beats the benchmark, and you validated it in mice, rats, primates, whatnot, at least currently, the buyers, they're not necessarily convinced immediately on the spot. They do care about what's the mechanism behind. And it's fair. To be honest, initially I didn't really understand why. So they said, what does it matter whether Avi dreamt about it at night or the AI created it? Eventually you have some molecule, and these are the results, and the results are beautiful and they're much better than the competition. So it's a go. No, people want to see the mechanism of action. People want to see the biology. And why? Because it's clear until you have proven models that are proven in translating into human, and until you have many of those, and until we have some kind of generation get used to it, you put bets on that. And you want to make sure that it's going to be translated into human, so you need to understand what's behind it. Then what we started to do at Mana is, for the short term, we are doing retrospect analysis. So on the things that are progressing into primates, etc., we are doing retrospect analysis to understand the mechanism and being able to explain it. And for the long term, we are working on explainability and mechanisms that are baked within the AI to being able to rationalize why the AI got to this result.
There's always a healthy skepticism about any new drug, especially now when AI is applied to complex biological systems. What keeps you guys up if there's something that bothers you that you're scared of, if there is any?
I remember when a drug that's now in phase 3, they injected it the first time to a patient. This is for treating osteoarthritis, and I was one of the inventors. I remember staying up the entire night. They say sometimes people, their entire life runs in front of them before their eyes. For me, it was each experiment ran in front of my eyes. And I asked myself, why did I go into this area of translational science? I should have stayed more in theory. A couple months later, when the data started coming in and we saw that we were improving patients' lives, it was worth it. At the end of the day, we're here to save lives. When you come to save lives and you have intervention with the people, there are a lot of issues that worry us. Once we do the science well and we work together and we try to troubleshoot it and we consult with a lot of colleagues and people that are critical in a good manner, I think it gives us also the conviction that we can bring something new to the table. And we are seeing that. It's like driving with a GPS. It brings us faster and more efficiently to the finish line. And if you ask me, Naresh, what really worries me, I came back from Germany on Thursday night and I met there an entrepreneur who worked for 20 years on a therapeutic, which was tested already in many patients, and they ran out of money in the middle of the clinical trials. Things became more expensive. This is a good drug, but they couldn't do it. That does worry me. We'll spend so much effort on the beginning and not be able to actually translate it to patients in the end. And I think AI is there really to help you save some of your funds. Also, not only that you do the science better and more efficiently, and you account for more challenges because the AI can read all the papers that are out there, but it's also about putting more effort on the patients themselves once we'll reach those clinical trials. That's the promise that I see, and that's what really keeps me awake, excited actually at night.
Fair worries. Anything on your end, Yogev?
I feel there is a huge opportunity here. When we started Mana, it was still kind of new AI in bio. It was not like a theme. Now it's a theme in every conference, every panel, every pitch deck.
Every dream.
That's the point. People kind of fantasize about it. Some people even put money, which is kind of hyped. And like I said, we need the funds. So one thing that worries me is that even though we do have the promise and the early signs of validation here, some companies would not succeed because of different reasons. And one thing one can be worried about and concerns me is that people will attribute failure to AI hype.
Just across the board and everything gets killed.
Exactly. Cut off all the funding sources for AI in bio, for platforms, and kill this route of innovation. And I feel AI is not a hype. In some areas, it's overhyped because people attribute to AI things that are not real, maybe. But there is a real breakthrough here that we have. It's a responsibility for every biotech and pharma leader to embrace AI in different areas. It's not a fit for everything, but in every area you can find something and look for the ways to use it, leverage it, and in order to accelerate science, development, and patient impact. That's a responsibility for all us leaders.
Absolutely. At the end of the day, it's being responsible. You both brought up money a couple of times. I have to get in there about your fundraising. You have a fantastic team. I think fundraising was relatively easy. That's what you mentioned earlier because you have previous successes. What is that experience like? If a novice founder, a scientist who has not started a company is trying to raise money, do you have any advice for fundraising for them? Walk us through your own experience with fundraising. You raised about $20 million of today.
So when we started, we got immediate raise from investors, Landlord and Unfix. I mentioned the investor in a previous company. That was a pre-seed. Then we raised the seed round, which was led by A16Z, Bio, Base 4 Capital. The internals joined, and the Technion also joined as participant, and some other angels from the field. So we raised a total of $20 million. We feel fortunate about the investors that we have and the board which is assembled. Everyone is helpful, positive, and excited about this journey. And they're all big believers in this AI and bio, at least for us, and with the vision of Mana. And we feel really fortunate. In terms of advice to newcomers to the field and scientists, the best advice is to go and seek advice. I'm not coming from deep scientific background, so what I say from my experience from maybe the other side, but when we started and when I came fresh from industry but not entrepreneurial journey, it's the same experience, which is you need to find the sources where to learn from, and who to learn from. And when we started Genome Compiler, which was 15 years ago, no one did really software in bio. It was not even a niche. It was really non-existing. So it was hard. And now you have many people that you can actually consult with and learn from their mistakes and from lots of my previous mistakes. So I'm happy to share it. And I do share it in different forms. As scientists, you are trained to learn. Entrepreneurship is yet another learning experience. So you need to learn. And every day, every week in the office, it's a learning experience. Sometimes there is a decision to be made. No one knows the right answer. Not me. Even not my board if I ask them. But somebody has to make a decision. So I make a decision, and in the mind, you need to think about it as an experiment. It has to be a controlled experiment. Don't bet the entire money in the bank on some experiment, some study. For sure not. But it has to be some controlled experiment. This is the way to look at it. And then learn from the results quickly. Be disciplined and be learning all the time.
Great points. Avi, any pointers from you?
When we started fundraising, Yogev and I used to go together and discuss the biology, discuss the chemistry, and the promise. Yogev spoke it so well and understood it so deeply that I told him, Yogev, it's better I visit the lab than I continue to go around and meet with these investors. You know, we were having sometimes some mornings 3 breakfasts. You have breakfast number 1 with investor number 1, breakfast number 2 with investor number 2, and breakfast number 3. And you need to pace yourself. Connecting with the investors is always about listening to their questions and understanding their fears. At the end of the day, they're giving you their money. They trust you, that you'll do your best to succeed, but they also want to see how this will materialize into not only great healthcare, but also into a great investment. For them, that's extremely important. I think I learned something from every one of those encounters, how we can be better. And many times through their questions, we went back and we said, that's a feature we need to add to our system. This is a concern they have. People are looking more about safety or more at targeting or another feature that we may want to add to our system, let's think about that. These are smart people out there. Many of them did not become rich just by chance. They became rich and successful because they were clever, and we can learn from them too. For me, the most important thing was actually, in some cases, to say also no to investors. And throughout my career, Yogev's career, we chose investors we know that will be with us also when times are great and also when times are hard. People that share the same values as we do. And I think that's a very important thing, because when things get pressed, you want to have good people on the ship together with you. And those investors really are with us. We've seen it time and time again. It meant a lot to us. Their support, their smart, their kind words, their insights that helped us. Sometimes they said we need to change a little bit the direction. We knew it was always in a great intention. We see them as partners, not because they put in money, but because they're our real partners.
I see a nice dynamic between you two. You trust each other. You trust Yogev to go out and do the scientific talk to. I think that trust is super important. And you have an interesting founding team. Two repeat entrepreneurs, Yogev and Roy Nevo, a world-class academic, you, Avi, and an AI pioneer, Kira Radinsky. How do you navigate decisions when the commercial and scientific perspectives might diverge? How do you balance those things?
It's something I learned from Bob Langer. Bringing your child to the kindergarten, you have to trust the teacher. In my eyes, I can advise Yogev. I can give my scientific opinions about specific matters that are within Mana, and I can also suggest new ideas in new areas. But I always remember that at the end of the day, what drives Mana is the business. Yogev, that's his expertise. He listens to me, but he has to balance in other things. He has to balance in the markets, the investment, the people he speaks to. He has a wider vision than I do when I come to look at this problem. I look at it through a very technological eyesight, and Yogev has to weigh in also the technology, but he also needs to think about marketing, future investors, the team that's working on this, in focus, of course. I really put my trust in the CEO. I may help navigate, but he's the captain of the ship.
The dynamic in the team is beautiful. It might be better than I imagined in the beginning, because like you said, Naresh, we are all, by the way, the four of us are repeat entrepreneurs. Roy and I more on the operational side. Avi has multiple companies out of his lab, and Kira Radinsky. We are all repeat entrepreneurs, and we all have things to say, and we all have credibility and strong opinions. It goes back to the ideation phase. As I mentioned, Roy and I prepared these cultural core values for the company to be. We didn't know what the company is. We didn't know who is going to be joining us. And based on that, we chose the partners. Luckily, Avi and Kira, they like-minded like us. It makes it much more smoother. And another point to assert it is that we are now 4 years into the company. Avi and Kira are scientific co-founders. They are not on the payroll. So people would expect Avi not to be involved in the company at this point. And actually, they are involved at least weekly, if not daily basis. And you see, super energized, super driven, work with the teams, tells you everything. Okay, now, in terms of how it works, I think everyone just respect everyone else and the other's opinions. It's not that we're not arguing. Of course, we need to argue because everyone has opinions. But eventually, everyone has its superpower, which the others respect. In terms of making tough decisions and areas where I'm not the expert on, we are lucky to have, as I mentioned, our investors and board super helpful and available whenever we need them. And we also have a pretty active scientific advisory board. And that's something which I would also advise a newcomer, like a scientist that is starting his own journey. Build some sort of trusted network around you. Our scientific advisory board are formal trusted network. We also have trusted network which are not officially that I speak with, but this is important. Surround yourself with people that can advise on different things. And then the next challenge is to know who to listen to and what advice to actually accept, because everyone coming from his own perspective. So that's important. And I do want to give a shout out to our advisory board, which are really amazing, not only experts in their fields, but also super excited about Mana and driven and forward thinkers and like-minded, and this is amazing.
Are there topics that we haven't talked about that we should have talked about?
We talked a lot about Mana. We talked about delivery, lipid nanoparticles, business strategy, fundraising, all of that. I think one important takeaway in this area of AI and bio goes back to the data generation. As a society, we generate a lot of data, each company and academia, all the experiments that are being done, just ton of data. Only a small fraction of it is being published, as we discussed, through papers and patents. I would urge everyone to try to find ways to share more data. There is a lot of data which is not considered necessarily proprietary and like the secret source of your company. I'm in discussions with some other leaders about how to make a case for everyone to share their kind of junk data. Think about junk data, because once junk data is good for another one, and it will benefit everyone. And this is outside Mana. This is just for the society, for all the people that use AI, for different models, for different types of molecules, modalities, whatnot. This will really accelerate science. This will really accelerate drug development. And I think this is something that we really need to take into considerations how to do it. In other industries, it happens. There are companies which are like infrastructure and foundation for different topics, and they get the data from their customers and share it and, train their models. It doesn't happen in the pharma industry, in biotech. We understand why, because data is precious, but I'm sure there is enough data that is not so precious that you can actually share, and that can be used by companies like Mana and others to leverage it and to really accelerate development. So that's some food for thought.
This very, very important point. I completely agree. I wish I could see some negative data so I avoid doing a mistake, avoid running an experiment, save time, energy, and heartache. Avi, any thoughts from you on that?
I want to thank you really for highlighting Mana's story, because when we started, people were less into AI and the design of lipid nanoparticles, of drug delivery systems. Today, it became mainstream. For me, I think looking at the future is really, where can we bring that first medicine to help patients the first time? Using AI to design it and working together with clinical partners to realize this dream. I'm optimistic it's going to happen sooner than later. Going back to your previous question, I think that the regulatory bodies, because you account for so much data when you come to design these systems using artificial intelligence, they may even have more confidence in these systems than less. It may take a bit of time to realize, but I think that's the way we'll look at it later on.
I want to close this with giving an opportunity to you guys to talk about one or two people that really, really made a company happen, or who you want to acknowledge. Those people are really important. Yogev, you want to go first?
One of the people that I can really highlight is one of our investors on board, Omri Dori. Omri Dori is a longtime friend and business partner. We've been together in Genome Compiler, so we know us from long ago. He was the one that initially introduced us to Avi. By the way, also to other people, but Avi was the perfect match. And he's also an active investor, board member, really, really, really wants this to happen. Good energy and lots of good skills, which we leverage every day. And I'd like to thank him. On that sense, I would also like to thank our other board members, investors, advisory board, the team, of course. We have a large network of supporters. Some of them are not officially engaged with Mana, and they're all contributed. So all the pharma partners, all different people that I talked to that kind of gave me advice or their thoughts, which are honest and objective, it really, really helped. And I want to say to everyone that at the end of 2025 now, we all hope for 2026 to be maybe a beginning of new era, not only AI and bio, but also the biotech in terms of the capital markets to kind of revive. We already see some good signals out there in the markets. And I think this is, again, an obligation for all of us to identify the right opportunities, take them, and accelerate them using support, funding, whatnot, in order to bring medications to patients faster and better. There are so many indications out there that needs delivery in our case, so many patients that wait for this medicine. So it's our responsibility. Let's go and make real patient impact.
Amazing. Avi, do you have any on your list?
Kira Radinsky, because she showed to me for the first time how AI can actually integrate into wet science, and together with her, the Mana team. Of course, my students in the lab teach me so much. Today, the focus is the Mana team, and I would say it's the blend of people inside Mana. They work day and night to make this dream happen. For me, every time I come in to the offices in Tel Aviv, it gives you a lot of energy. And I hope, Naresh, when you come and visit Israel, you'll feel that energy. As you enter, it's something special. And it's that 1 and 1 really equals far more than 2. And that's what you feel actually when you enter. So I want to thank all the people that contribute within Mana, and Yogev, of course, you for leading this venture. It's been a really, really fun journey. And I look forward to many years together.
Avi and Yogev, thank you so much for sharing your story. I think this is my first episode where AI and bio are involved. So I learned a lot. I'm sure my audience is going to appreciate this. I hope we can do a next round. When you make some more progress, probably we'll do a live one in Israel if we can. But good luck and Godspeed to you guys.
Thank you so much.
Thank you so much, Naresh, for the opportunity. A special thank you is also to Roy Nevo. Been doing this journey for a while, so we go back a while together. And at Mana is really the magician. I do the speaking, I do the presentations, I speak on stages, but Roy is the one that actually make everything I spoke about a reality.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.